Hi everybody, it's me, it's Dr. Kestler from Lorain County Community College with today's co-video. Today is April 27th. To begin with my temperature, I am 96.7 and 35.9 Celsius. And it is a beautiful day outside, so I am enjoying the outside today because it's been a long time that we have been out. We are getting ready in Ohio, as most of you know, to go back to, or to emerge out of a lockdown, if you will, or whatever it is, this closing that we've had. And I want to just remind you, I'm just going to refer to the co-video from Friday. Go back and look at that. We all need to be very good and document when we do, and if and when we do get sick. We need to be very careful about our steps and be very thoughtful and mindful so that we don't end up closing up again. I spent Saturday in a conference. The conference was actually from my living room, but I was listening to people from Dr. Fauci from the NIH. I was listening to the CDC director from China, as well as a drug developer from England and a vaccine developer from the United States. And it was a fascinating, and an NIH scientist as well, who taught us a little bit about the biology of the virus. And I want to talk a little bit about that, about COVID-19 as a virus. First of all, it has the following kind of a structure. Shown in this little cartoon here. There is an RNA, which is quite large as a chromosome. It has a spike protein. Those of you who've had me for class know that I prefer to say viral receptor binding protein because it's a little bit more descriptive. And it has it's an envelope protein that has a membrane that is coming from the host. And it has what we call a positive sense RNA. The replication cycle is somewhat complicated. There is an RNA enzyme, replicase, that is used, and this is the life cycle here, shown here, and then it gains entry. This is a non-nuclear virus. So those of you who know what I'm talking about, there's the nucleus and the cytoplasm. This is a cytoplasmic virus. And it goes through replicating lots of pieces and parts that eventually get assembled into viruses that get released from the cell. One of the problems that we have in developing drugs, I'm going to talk a bit about drugs and vaccines today. That's going to be a big part of what we're going to talk about. One of the big problems that we have in developing drugs, for instance, as well as a vaccine, is that we have to be cognizant of a little bit of a peculiarity of this particular virus and other coronaviruses like it. And that is, there is two ways that the virus can get into a cell. Sort of a front door and a side door, if you will, and that is kind of a unique problem, is what that is. That means you have to control both. You have to prevent both, and so there's a process called endocytosis where it gets engulfed or swallowed up by the cell, sort of like a Trojan horse. A mistake. You don't want to take that inside, or you take it in and through a receptor. So there's a fusion of events, and so we have to figure that out going forward. All right, what I want to do now is, so that's a little bit about the biology. There was actually a lot more, but probably only interesting to me. I filter away a lot of the stuff that you guys might not want to listen to. But I do want to talk about vaccines. And there are vaccines that are in explore. There's like 107 vaccines being developed right now. And that's a great thing. And we need them. Let me tell you when we need them. Yesterday. We need a vaccine yesterday. And so in order to get this vaccine, we have, first of all, you do the exploratory part where you're looking in the laboratory, maybe even animal models. You're doing that kind of work first, and then you go into clinical trials, preclinical trials, then clinical trials, and then you find one that hopefully works and does the best job, and does not cause any problems. So that's where we are on this. We have a lot of candidates. We have some that are actually in clinical trials right now, and it's a very exciting thing to be thinking about. But we need to be very, very fast on this. And the reason that we need to be very fast on this, because we have over 3 million people infected with this virus. And certainly not at this time, but very soon, by the end of today, there will be 1 million Americans. That will be 1/3 of the infections will be in the United States. And we need to get moving really soon on this vaccine. And we really have to do something, since we need it so fast, we cannot wait for a long period of time. So we need to take 3 steps. Normally we go through very laborious steps to make a vaccine. We don't have that luxury. We need to build capacity to develop, test, manufacture, and distribute lots of different candidate vaccines, even though we may only want one. Why is that? Again, we need this yesterday. We need a vaccine as soon as we possibly can have one. So we have to be, as we're building, we need to be building manufacturing. Now, this is going to be costly because normally you have a competition, and then whoever wins builds the factory. Nope, we need to build them all simultaneously, or at least partially, so that we can get this thing scaled up to size really quick. There will be a shortage of vaccine when it's first made. That is always the way. That's what happened in 2009 when we had the H1N1 vaccine for flu. We gave it to the places where they needed it first. It wasn't readily available. And so we also have to build a distribution system so that we can get it out everywhere. One of the things that we have to be very aware of is other people's diseases are our problems in America. That is just the way it is. That's just a fact of life. And we best get used to that. And plan. We need to, if I might be a little bit forethinking here, I want to think about not this current pandemic, but the next. We need to build this kind of a system so that we are ready for when and if it happens again. But I think it almost surely will happen again. All right, so there are many features that are conserved in so I'm talking about drugs for a second here, and we're just going to, in general, there are many things that you can go after, and I want to point out something that I was listening to one of these talks, and it just struck me hard, in that there has been a vaccine that's been worked on for the first SARS virus, SARS-1, and that virus and MERS, they were working on those too, but those were abandoned because, well, it wasn't big enough of a problem. I think we should have kept going on that. We should keep going on all kinds of avenues of all kinds of infections. I think that those of you who are young, looking for something to do in life, become a virologist. We're going to need you. We're going to need you desperately because we need to be ready for the next kind of thing. So there's a lot of pathways. There's a lot of pathways that are present that are host protease dependent. There are messages that are in common between the different viruses, and enzymes that are common between the different viruses. And I'm talking about SARS-1, MERS, and COVID-19, the SARS-2 virus. And I want to just point out to you in this next figure here, they've been working on a potential target for a drug that targets all 3 viruses. So had we been working harder on SARS-1, we may have had something in place very soon for SARS-CoV-19, COVID-19, the second SARS virus that came through. And so I guess that's my point in all of this, because there is this one compound that looks like it's promising. And of course, as you are all learning, developing drugs is not, just go do that. No, it is a very, very labor-intensive process where you have to put things together. It may, in fact, be the case that you need multiple drugs to target. Like in HIV, we use 3 drugs to target HIV. That's why we're so successful in targeting HIV. We may need 3 drugs to target it, and we need to figure out which ones of the 3 will do the best to ameliorate the disease. And so that is a lot of work, and people will be going after that. I'm going to leave today with one story, and this is one of those helpers things. You know, there's a lot of people who are tired of being locked up indoors. I'm outside today. I am too. I like to get out. I like to be around people. I like to talk to people. I like to as well. I want to, but we are in an effort here. We really are. And I wish there was a shortcut, but I'm telling you that the lockdown has worked. It has been very effective, and hopefully we will emerge from it, and we will be able to control it as we emerge out of lockdown. It will be different. But I want to tell you a little story here that this was just a great story. There was a company in Pennsylvania where a group of, a couple dozen workers got together, and they realized they were making PPE, and that's the equipment for protection that the people, personal protection equipment, and they realized that if they were to go home, they might get sick and they might have problems. So they decided to stay in the factory, live there 24 hours a day, live in the factory with each other, because they knew that no virus could come in as long as they were in. And I just think that's a great story, and it reminds us all to be good to each other. That's what this is all about. This is about being kind to one another, simply put. And may you all persist like a lentivirus. Thank you, and have a beautiful day.